Hello, everyone, and welcome to today's webinar, Next Generation Flow in Multiple Myeloma Chemical Trials. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by BD Biosciences. To learn more, visit www.bdbiosciences.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you're experiencing any technical issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speaker, Dr. Bruno Paiva. Dr. Paiva, you may now begin your presentation.
Thank you very much for the kind introduction, as well as for the invitation to participate in this webinar and to talk about next generation flow in multiple myeloma clinical trials. This is a disclaimer. These are my disclosures, and this is the outline of my presentation. I'll first talk about the International Myeloma Working Group MRD criteria that was published back in 2016 and will probably be updated this year. Then I'll make the case for the use of NGF, also showing evidence from clinical trials using this method. Then I'll also talk about the FDA ODAC hearing that took place in April last year and led to the approval of MRD negative CR as an endpoint in clinical trials. And finally, I will talk about the transition from clinical trials into routine practice. These are results shown a few months ago at ASH by Dr. Noemi Puig from the Spanish Myeloma Group. This is very important because once again, we show here the lack of prognostic value associated with the complete remission status in transplant-eligible patients enrolled in different Spanish clinical trials. And regardless of whether you look to the moments after induction, transplant, consolidation, as well as during maintenance, there are no statistically significant differences in terms of progression-free survival between patients achieving CR versus those who did not achieve CR. Now, in the same meeting, Dr. Puig also showed results from transplant-ineligible patients enrolled in a different Spanish clinical trial. And while in this study, it is true that the immunofixation status that ultimately defines complete remission after 18 induction cycles was prognostic for PFS. Once again, if we focus on patients with a negative immunofixation in complete remission, there are significant differences in PFS according to the MRD status in the bone marrow assessed by next generation flow. And once again, we see that those patients with positive MRD have a PFS that becomes quite similar to those that did not achieve a complete remission. This is why it was so important back in 2016 to redefine the CR criterion in myeloma by adding MRD negative definitions. There was a sequencing MRD negative definition that required CR plus negative MRD with a sensitivity of 10 to the minus 5 using the clonoSEQ platform, as well as a flow MRD negative CR that required the absence of phenotypically aberrant clonal plasma cells by NGF on bone marrow using the EuroFlow standard operating procedure using 8-color 2 tubes for MRD detection with a minimum sensitivity of 10 to the minus 5. There have been, I would say, extensive debate about the similarity and concordance between NGF and NGS for MRD assessment in myeloma. There were multiple studies published in the last 5 years. I chose one that I think is representative of all the data that has been published. This is data from the KARMA-1 study, the pivotal study that led to the approval of one anti-BCMA CAR-T product, ide-cel. And in this study, NGF and NGS were performed simultaneously in a large number of samples from more than 100 patients. And overall, you can see that the concordance is generally high, and it ranges from 63% at month 1 after infusion up to 81% at month 6 after CAR T cell infusion. And in a sub-analysis within this study, we showed that discordances were not that much related to the fact that one method was showing positive MRD and the other was negative. By contrast, discordances were mainly associated with hemodilution that is very frequent in the first few months after CAR T infusion. And in fact, discordances were mainly those samples being classified as hemodiluted by NGF, whereas using NGS, these were being classified as undetectable MRD. Therefore, NGF does offer the possibility of looking at hemodilution in real time and therefore avoid a potential false negative result, and also allows for the possibility at looking to the bone marrow cell composition and eventually find other aspects that may be prognostic. And again, in another example from the same study, the KARMA-1, we showed that among patients with negative MRD, PFS was different in those with no normal plasma cells and those with reappearance of normal plasma cells. In other words, the reappearance of cells expressing BCMA is probably a surrogate of either loss of CAR T cell persistence or, in the case of persistence, loss of CAR T function and, in consequence, inferior PFS. In addition, there is an imaging MRD negative criterion that requires CR plus MRD negative as defined previously by either NGF or NGS, plus disappearance of all areas of increased tracer uptake observed by PET-CT. And this is quite important. Perhaps not that much in newly diagnosed patients, but definitely in those with relapsed refractory disease in which the probability of extramedullary disease is higher. And again, in the same trial, in the KARMA-1, we showed that among patients that were MRD negative in the marrow by NGF, PFS was different in the case of absent versus present extramedullary disease detected by PET-CT. Finally, and in my opinion, the most important criterion is the one of sustained MRD negativity confirmed at least for 1 year apart. And I think that sustained is critical to show reproducibility of either an MRD negative or positive status, and this will better stratify patient survival. Here we show the results of sustained and detected MRD for 12 months using NGF and here using NGS. And as you can see in the slides, the results are nearly identical. And this is why I believe, and I believe there is also consensus at the international level, that nowadays NGF and NGS are considered interchangeable MRD methods in myeloma. Now, making the case for NGF, I think that overall MRD assessment in myeloma using NGF has been clinically proven. It is highly sensitive and it is an integrated solution, as I alluded before. There are many advantages as well as some limitations. Regarding advantages, it is a fast method, only 3 to 4 hours from sample reception to data report. It is ultra-sensitive with a limited detection ranging from 10 to the minus 5 to 10 to the minus 6. There is end-to-end standardization using physician-developed protocols and ready-to-use panels with clinical utility. It is integrated and reproducible and includes automated analysis and reporting using clinical expert-developed databases. And these same databases allow for external and internal quality control in a per-sample basis. As I showed before, NGF provides information on normal and malignant cells, which is very useful to assess sample quality and the reconstitution of normal compartments. It may help in the identification of MRD targets for next immunotherapy, and from the research point of view, it also allows for purification in real time of MRD cells for subsequent characterization using multi-omics. Regarding limitations, it obviously requires a fresh sample, ideally in 24 hours or less. Despite the use of automated analysis and databases, it requires education and training, let me say, as any other method used in laboratory diagnostics, and many cells are needed for the required sensitivity. For example, 10 millions if you want to achieve a limited detection lower than 10 to the minus 5. Our experience is that it works, and it works in virtually all samples. This is data published already a few years ago, probably the first Spanish clinical trial in which we used NGF, and MRD data was obtained in virtually all samples. The median limit of detection was approximately 3 per 10 to the minus 6, and a minimum sensitivity of 10 to the minus 5 was achieved in virtually all samples. Now, what is the evidence from clinical trials using NGF? And I must say that fortunately there are numerous clinical trials with reported data using NGF. In sake of time, I will show only one or two examples, very recent ones. This is data published by the German group, Elias May, a few weeks ago in the Journal of Clinical Oncology. NGF was used for MRD assessment in the GMMG HD7 clinical trial, a phase 3 study that randomized newly diagnosed transplant-eligible patients to the standard of care, VRD, versus the experimental arm, VRD plus 1 anti-CD38 monoclonal antibody. And after transplant, there was again another randomization into maintenance with lenalidomide or lenalidomide plus isatuximab. And I would like to stress out that the primary endpoint of this study was the post-induction MRD negative rates using NGF at 10 to the minus 5 together with PFS after the second randomization. These are the results of the most recent publication, and here in the left part of the slide you can see the clear prognostic value of NGF in this study, MRD negative, MRD positive patients with a hazard ratio of 0.4. In other words, a 60% increased risk of progression and or death if patients were MRD positive after induction. And I think it is very important to recognize that within MRD negative patients, there are no differences regarding treatment randomization into the 3 or the 4 drug regimen. In other words, it is more important the achievement of a deep negative MRD rather than the treatment itself, although we know that the 4 drug regimen led to significantly higher rates of MRD negativity. This is very similar data from the same trial, but now using the concept of undetected sustained MRD from induction throughout maintenance. And as you can see in the slide, those patients with undetected sustained MRD are experiencing a PFS that is close to 90% 3 months after, 3 years, even 4 years after the beginning of maintenance therapy. These are very promising outcomes. And once again, outcomes that depend much more on the achievement of sustained and detected MRD rather than the treatment that led into that very sensitive, deep, and durable remission. I'll now talk about the ODAC hearing that took place last year at the FDA. As a way of introduction, I think that if we look at the relative survival at 5 years based on the year of diagnosis, namely the evolution in the myeloma treatment from steroids to alkylators, transplant, the former novel agents, proteasome inhibitors and immunomodulatory drugs, more recently monoclonal antibodies, and even more recently the newer immunotherapies. And if we look to the cumulative rates of undetected MRD that continues to increase with each new treatment option, despite the fact that the sensitivity of the methods also increased over time, I think that overall this slide illustrates how the efficacy of new regimens, deeper and durable MRD responses, and prolonged survival are interconnected in myeloma. We are truly living in a unique time in progress of myeloma therapy, with 19 drugs approved in the last 10 years that obviously led to a significant prolongation of survival outcomes. Fortunately, there are multiple new drugs and combinations under evaluation. Unfortunately, patients will have to wait for longer and longer periods for documented PFS benefit or the observation of a lack of survival benefit. And this is why it was so important to have MRD negative CR considered as an intermediate endpoint for accelerated approval of new treatments in multiple myeloma. This was achieved, and I should say unanimously, because at the FDA ODAC hearing we showed a very strong individual patient level correlation between MRD status and progression-free as well as overall survival. And we also showed a promising trial-level correlation between the treatment effects on the MRD negative rates and the treatment effect in PFS as well as overall survival. In other words, if we look into a randomized clinical trial, and if in that trial the differences in MRD negative rates are, let's say, 15% or greater, this will most likely predict a clinically meaningful improvement in progression-free survival. Here we are comparing the difference in MRD negative rates between the experimental versus the control arm in clinical trials, those with a hazard ratio of 0.6 or more, and those with a hazard ratio of less than 0.6. In other words, a clinically meaningful improvement in PFS. And as you can see in the slide, the differences in MRD negative rates are clearly different. There is almost no overlap between meaningful and less meaningful clinical trial results. This is using MRD for learning in real time. And to exemplify what I just said, I will show one study that is out of the press. It was published last week or 2 weeks ago, maybe. This is the comparison of MRD negative rates on the intent to treat population 10 to the minus 5, in the Cassiopeia trial, in which patients were treated with dara-VDD, versus the MEDIS trial, in which patients were treated with isatuximab KRD, both conducted by the French myeloma group. And as you can see in the slide, MRD negative rates almost doubled from one trial to the other, 35 versus 63%. This is knowing in real time how much benefit we can potentially obtain with a new induction regimen. Not only that, in terms of cross-study comparison, but also learning in real time within one study who are the patients that are benefiting the most from the new regimen and who are the patients that may not benefit that much from this specific regimen. And in this slide, you can see how patients with a 414 translocation, that is a high-risk cytogenetic alteration in myeloma, do benefit, achieve very high MRD negative rates, and the same does not apply for those patients with deletion of 17p, that is another high-risk cytogenetic alteration in this disease. We know that patients with very aggressive high-risk myeloma may not achieve the same rates of MRD negativity, but I think what is most important is to have a better understanding of what is the impact of achieving a deep and durable MRD negativity in these patients with very poor prognosis myeloma. And a few years ago, we showed again in one Spanish clinical trial that those patients with high-risk cytogenetic alterations achieving deep and sustained MRD negativity by NGF with a sensitivity of 10 to the minus 6, sustained for probably 3 years since the induction up to 2 years of maintenance therapy, these patients showed the same PFS when compared to those with standard risk cytogenetics also achieving undetectable MRD. Now finally, clinical trials and routine practice. And it is true that despite the cumulative positive evidence of MRD assessment in clinical trials in myeloma, there is still reluctance and some controversy regarding its use in routine practice. There are some arguments. I would say that probably the most frequent are these two. I've tried, but the results are not reproducible. Secondly, it requires a bone marrow aspirate. Now, regarding the first argument, it should be recognized that the efficacy and duration of treatment will have an impact in the survival outcomes of patients achieving MRD negativity, particularly if differences between treatment are very high. In this slide, we show a pooled analysis of 4 different clinical trials. And here we are seeing PFS of patients achieving MRD negativity in the experimental versus the control groups in these clinical trials in the blue curve. And the differences start to be significant after 3 years since randomization, because the experimental arms were clearly superior and the quality, the depth of MRD negativity between the data and the control group is different. In other words, if the treatments being used or available in routine practice differ significantly when compared to those in clinical trials, the depth, the quality of an MRD negative result may not be comparable, and therefore survival of an MRD negative patient will also be different. The other point is methodology. If we don't use the same methods that are being used in clinical trials, particularly those endorsed by the International Myeloma Working Group, we cannot guarantee the same results in clinical trials and in routine practice. By contrast, if we use the same methods, then the results become quite similar. And as shown in this example, using NGF in a clinical trial and in the routine practice of the Greek myeloma group. Another important aspect is the predictive value of MRD. And nowadays we know that the predictive value of MRD increases quite significantly if we leverage on MRD kinetics instead of a single MRD assessment. This is data from, again, the Spanish myeloma group, leveraging in almost 1,800 MRD assessments and using the connector algorithm. We stratified patients into those with sustained MRD negativity, excellent PFS and survival outcomes, those with detectable but stable MRD levels, slightly inferior PFS, interestingly the same OS, and then patients with detectable and evolving MRD levels that display dismal overall survival. And from the statistical point of view, obviously MRD kinetics are of independent prognostic value together with staging and diagnosis, but I would say that more importantly, if we look into the predictive value, the C-index, MRD kinetics are clearly superior when compared to a single MRD assessment, for example, before maintenance, and clearly superior compared to any risk model established at diagnosis. Then the second argument, it requires a bone marrow aspirate. And this is true, and this may be inconvenient to many patients, particularly the elderly and the frail patients. This is why it is so important to present, to investigate, and fortunately to be able to present some promising, preliminary yet promising data on the concept of monitoring peripheral residual disease using NGF. Again, out of the press, this was published a few weeks ago in the Journal of Clinical Oncology, another study from the Spanish myeloma group, in which we show that it is possible to detect peripheral residual disease, PRD, if you may, CTC, circulating tumor cells, in approximately 10% of transplant eligible patients during maintenance or observation. And this is associated with significantly inferior PFS and OS. Does it mean that because NGF is so sensitive, we can do blood and forget about the marrow? The answer is no. That's not the point. The point is to provide additional information in between different bone marrow aspirates. And in this slide, we show the prognostic value of simultaneous monitoring of PRD in the blood and MRD in the marrow using NGF. And as you can see in the slide, there are three different patient subgroups. Those with double negative results, excellent survival outcomes, those with double positive, dismal PFS and OS, and a third subgroup of patients that was negative in the blood, positive in the marrow, and the PFS is inferior when compared to those that were negative in both samples, meaning that the marrow is still the gold standard specimen, and a false negative result in the blood will have consequences in terms of PFS. On the other hand, I would like to stress out that in a patient with positive MRD in the marrow, if you want to know if the patient is at risk of progression in 3 years' time, or by contrast, in the next 6 to 12 months, then looking at the blood is very informative. And if you see disease in the blood, then you know that the risk of progression in 6 to 12 months is extremely high. We can improve on our ability to detect disease in the blood almost to levels identical to the marrow when complementing NGF with mass spectrometry. And again, here we are seeing how using these two methods we obtain PFS and OS curves almost identical to those we were seeing before using NGF in the blood and in the marrow. And this is because mass spectrometry is very sensitive and allows for the detection of residual disease in the serum in patients otherwise negative by NGF. And once again, NGF is providing complementary information to have a better understanding of patients that are at risk of ultra high risk of disease progression in the next few months. Based on this data, we will continue investigating to accumulate more data that could potentially confirm what is currently an hypothetical possible algorithm to test MRD in the marrow and PRD in the blood. And I would say that the bone marrow is the gold standard specimen for the high sensitive assessment of treatment efficacy, particularly during the first year of treatment. Such an approach would be aligned with the FDA ODAC MRD negative CR endpoint that takes place in between 9 to 12 months after treatment initiation. We also know that tumor burden in the blood is likely undetectable with a very effective current standard of care during the first year of treatment. And regarding immunofixation, or if you may, mass spectrometry that is more sensitive, the M component, the monoclonal immunoglobulin, long half-life may lead to false positive results early on. After this first assessment during the first year, then obviously to confirm sustained and detected MRD in the marrow with tailored periodicity regarding treatment as well as patient risk features, and in between, let's say for example every year or every 2 years, you may consider PRD assessment using, for example, NGF, not forgetting also the value of imaging modalities such as PET-CT. Then, if MRD becomes feasible, attractive, and informative, meaningful in routine practice, how to use it. And I will end my presentation with one example, and it is the use of MRD for discontinuation of maintenance therapy. There is more and more data. This is data published in October last year in the Blood Cancer Journal from the investigators at the University of Chicago, the MRD to Stop study that investigated discontinuation of maintenance guided by multimodal MRD negativity. And here I would like to stress out that in those 47 patients that were enrolled into this concept study, after a median follow-up of almost 3 years, the 3-year PFS was 85%, very promising. And some food for thought in terms of the future, not the present. It is interesting to see that those few patients that lost the MRD status or show a disease progression were patients with ultra low levels of MRD in the 10 to the minus 7 logarithmic range prior maintenance cessation. Data presented at ASH from the Greek myeloma group showcasing how sustained MRD negativity for 3 years using NGF may guide discontinuation of lenalidomide maintenance after transplant. This was not a clinical trial. This was an observational study in the hematology department from Athens, the University Hospital of Athens. And using this strategy, you can see how the treatment-free survival rates at 1, 2, and 3 years after stopping maintenance were very promising, 94, 92, and 76%. These results are very similar to those that we reported a few years ago in the Spanish 2014 maintenance study. This might have been one, if not the first, study using MRD assessed by NGF to guide the duration of maintenance. After 2 years of maintenance, different treatment arms that led to no differences in survival outcomes. We can forget this part of the slide. After 2 years, patients that were MRD negative using NGF stopped maintenance. Those that were MRD positive continued for 3 additional years with lenalidomide, dexamethasone. These are the results that were published by Dr. Rossignol a few years ago in the Blood Journal, and in my opinion, the results these findings provide three important messages. Number one, MRD is such a powerful prognostic factor that even if you stop treatment in MRD negative patients and you continue treatment in MRD positive, there are still highly statistical significant differences. Number two, patients that were MRD positive continuing treatment using the same drugs that were given during induction and consolidation and 2 years of maintenance may not be enough to overcome MRD resistance, and we need to think differently on how to tackle persistent MRD after such a long treatment duration. Third message, MRD negative. The 4-year PFS was 83%. This is very, very promising to consider safe treatment-free intervals in some patients. And the reason I say some patients is because there were a few that showed early disease progression after stopping maintenance because of an MRD negative result. And this is why now we are so interested in finding red flags of unsustained MRD negativity. And I'm happy to say that last year, in a collaboration with the Italian Myeloma Group, we showed reproducible results of what could be the predictors of unsustained MRD negativity in transplant-eligible patients. And these are an ISS3, the presence of more than 0.01% circulating tumor cells, the presence of 2 or more high-risk cytogenetic alterations, and a late achievement of MRD negativity. If patients show 2 or more of these red flags, then maybe stopping treatment because of negative MRD may produce suboptimal results. I always end by saying that there is no precision medicine without precision diagnostics, and I do believe that MRD assessment in myeloma will be paramount for that precision diagnostics and precision medicine. Definitely in the bone marrow, more conventionally, and probably in the future more and more in the peripheral blood. I'll stop here. I would like to thank again BD for organizing the webinar and the kind invitation, and I would like to thank you for your attention.
Thank you, Dr. Paiva, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located in the lower left corner of your screen. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question asks, which translocations are notorious for MRD?
Thank you for the question. I'm not sure about the meaning of the question, though, I must confess. If the question is related to what are the translocations and the cytogenetic alterations that may impact on the prognostic value of MRD, I would say that eventually those patients with deletion of 17p, even though achieving negative MRD, may be nonetheless at higher risk of disease progression. This has been shown, for example, in some of the French clinical trials. And again, this is why I believe it is so important to define a negative MRD result with the highest possible sensitivity, ideally 10 to the minus 6, and based on multiple assessments to define sustained MRD negativity. Maybe with a deep and durable 10 to the minus 6 MRD negativity, the outcome of patients with deletion of 17p could be significantly improved. As for the other translocations, nowadays I think that the outcome between or among MRD negative patients is more or less the same.
Wonderful, thank you. All right, our next question here asks, could you share some insight on the impact of MRD negativity on patient outcomes, particularly in terms of progression-free survival and overall survival?
Well, I think that the presentation was clear on that regard. I think it is fair to say that MRD is clearly of independent prognostic value for PFS and OS. If there were doubts, this was unequivocally shown in the largest ever meta-analysis conducted that was presented at the FDA and concluded a very strong individual patient level association between MRD and PFS and OS. And I should say that according to the experience of many groups, usually MRD is even the strongest prognostic factor above or beyond cytogenetic alterations or cytogenetic risk in myeloma. And if I may add, I think that this concept applies to all disease settings, newly diagnosed, transplant eligible, ineligible patients, as well as nowadays in relapsed refractory disease.
Great, thank you. All right, our next question here, we have a ton of wonderful questions coming in, so please keep submitting them. Thank you. Our next question asks, what are the main challenges in implementing MRD assessment in clinical settings?
Well, this may depend on the origin of the question, the geographical origin of the question, but I think that mainly it is access and partially associated with that reimbursement. I think these are the main challenges for a broader MRD implementation. And I think that the time is now to probably add some extra pressure on the health authorities based on data, particularly real-world data, in addition to clinical trials. I think that showing reproducibility between trials and the real world would be very important to confirm that MRD assessment, particularly using NGF, is also meaningful in the routine setting and therefore should be fully reimbursed.
Okay. All right, our next question here asks, what do you think about MRD by NGF for patients who receive bispecific antibodies?
This is a relevant question, thank you, because obviously the mode of action of bispecific antibodies, also CAR T cells, is different from the other agents that have been used for the treatment of patients with myeloma. This said, there is strong evidence showing that MRD by NGF is of prognostic value. One is the fact that in phase 2 studies, the PFS achieved by these agents, which is usually in the range of 1 year or slightly more, in a very heavily pretreated population, is associated with deep, durable responses. So again, there is an association between achieving MRD negativity and the benefit of these agents. And secondly, I think in January this year, we published a new study in the American Journal of Hematology showing clearly the prognostic value of MRD by NGF in patients treated with CAR T cells and bispecific antibodies. And again, in a multivariate analysis, it was the strongest prognostic factor.
Wonderful. All right, moving on to our next question here. This question says, for monitoring MRD in PB by NGF, can you follow the same Euroflow protocol? And what is the minimum volume of PB? Can the Euroflow database be used?
Yes, this is a very good practical question. Indeed, we use the very same Euroflow protocol. Just requires a little bit more of volume, 5, 7, 8 cc's of peripheral blood. I would recommend the collection of a 10 cc tube for MRD assessment in the peripheral blood by Euroflow NGF. And again, because the procedure is exactly the same, and basically all cell types are represented there with the exception of mast cells, erythroblasts, B-cell precursors, and some myeloid precursors as well. The databases can be generally used as well.
Excellent, thank you. All right, our next question here asks, is the hemodilution a characteristic in samples of patients treated with CAR T cells?
Yes, there is a unique feature of CAR T therapy and happens more frequently at months 1, 3, eventually 6, but definitely between month 1 and 3 after CAR T infusion. And obviously this is unrelated to the method. In fact, as I alluded in my presentation, if any, it highlights one of the advantages of NGF, which is to check for those hemodiluted samples. It is probably associated with the lymphodepletion prior CAR T therapy, as well as some impact of CAR T expansion in the bone marrow milieu and the differentiation of hematopoietic progenitor cells. Again, if any, this also illustrates how we should investigate what is the value of MRD assessment in peripheral blood in those first weeks, months after CAR T infusion, because eventually it may be more informative and definitely more convenient to the patient when compared to bone marrow aspirates.
Excellent. All right, our next question here asks, can NGS help monitoring MRD with NGF in ADC therapies? And what other scenarios may exist where NGS would be preferred over NGF?
Well, I assume Zoltan is asking for the impact of antibody drug conjugates and eventually belantamab, which is approved for the treatment of relapsed refractory myeloma. I think that in this kind of therapy, with this kind of therapy, there is no difference whatsoever between NGS and NGF in terms of the ability of these methods to detect MRD during or after treatment. Therefore, I see no impact, again, of these therapies on the efficacy of both methods. And as for the scenarios in which one would choose one method versus the other, again, this would be very study specific or geography specific. But there are some examples. In very large multi-center international cross-continental clinical trials, NGS has the advantage of allowing for storage, freezing, and batching of samples, which is more difficult, obviously, with flow cytometry that requires a live sample, fresh sample to be analyzed in 24 up to 36 hours. Whereas, for example, if this is a clinical trial, let's think again on an example with belantamab. If this is a clinical trial in relapsed refractory myeloma, or a clinical trial exploring the drug, for example, during after consolidation, in which you may not have the baseline sample to calibrate for NGS, then NGF would be the method of choice. The main message is that these methods are, in my view, interchangeable. They are both well standardized, optimal for MRD assessment, and you can choose one or the other based on the specificities of your environment.
Excellent, thank you. Our next question here asks, how is currently measured MRD with CAR T cells therapy?
Well, again, this is an open question because there is still limited data on MRD on CAR T cells. In fact, only a few studies have been published. One was extensively described during the presentation. Again, I would like to point out that there are already two phase 3 randomized clinical trials comparing CAR T versus standard of care in early relapsed refractory myeloma, in patients with relapsed refractory myeloma. the first 1 to 3 lines of therapy after disease progression. And the added value of CAR-T versus the standard of care is well shown both in the difference between MRD negative rates and PFS. Again, there is this strong association between treatment effects on MRD negative rates and PFS, and this holds true also in CAR-T therapy. As for, from a more practical point of view, as for the time points, honestly, I don't know. I think we still need more data. I think that 1 to 3 months after infusion, ideally in the blood as opposed to the marrow, would probably be a sign of primary resistance, and therefore the physician could be alerted for a need to intervene sooner rather than later. And this is particularly relevant because at that time, the M protein is probably not that informative because of the long half-life that will not clear up until 6, even 9 months after CAR-T infusion. And then I think that the 1 year response, the sustained MRD response at 1 year, is informative to identify those patients that at that specific landmark continue to be at risk of progression, or by contrast may potentially enjoy long-term PFS with a single infusion of CAR-Ts. I think this is very important, particularly from the patient perspective.
Wonderful, thank you. Our next question here, any association between immunophenotypes, aberrancies, and MRD positivity?
Not sure about the meaning of the question, but I will hypothesize that the question is about if there are immunophenotypes that are more difficult to monitor. Well, I think this is true. Obviously, there are patients with myeloma in whom the aberrant plasma cells have a myriad of aberrant immunophenotypes, and in others, these are more limited. What is my experience is that with the 2 8-color panel of HeroFlow NGF, you can identify at least one aberrant immunophenotype in virtually all patients. One important aspect is that there are some patients that, for example, a diagnosis may not present with myeloma cells in the marrow, for example, those with macrofocal disease. But besides it, I think that the 2-2 strategy, particularly with the kappa lambda as well, usually allows for identification and monitoring of all aberrant immunophenotypes. Another relevant aspect is that even though it is not as frequent as in other hematological malignancies, for example, AML, we do see occasionally some shifts in the aberrant phenotypes. Therefore, in my opinion, the approach should always prioritize the different from normal concept rather than following only those aberrant immunophenotypes identified prior treatment.
Wonderful, thank you. All right, moving on to our next question here. This question asks, is there a quantification method or mechanism that helps in confirming relapse? Like, is there a threshold where the MRD stops being MRD and is now a relapse?
This is a very interesting question. I'm glad we do have some time for discussion. Well, the answer to the question will evolve because of how endpoints in clinical trials and outcomes will be measured. And it may well be that in some future clinical trials, MRD reappearance or an increment in MRD log levels, for example, 1 or 2 logs, will be considered as an MRD progression, a molecular progression, and this may trigger early rescue intervention. There are clinical trials that are ongoing exploring this concept. This said, the other side of the question is that an MRD positive result is systematically associated with inferior survival. However, the time between positive MRD, and even MRD reappearance, and disease progression may vary considerably across patients and across treatment protocols, and may well be 3 to 4 years. Now, if you are considering early intervention because of positive MRD, 3 to 4 years until disease progression, particularly off treatment, may be considered too much, because you would be starting a new line of treatment and preclude a 3 to 4 year treatment-free interval to the patient, which obviously is very convenient from the quality of life point of view. This is why it is so important to see over and over again, and we have now a new study that will be submitted soon, very soon, showing that among patients with positive MRD, if you want to know if the risk of relapse is imminent, or by contrast, it may take place later on, looking at the blood is very informative, because if it is positive in the blood, then usually patients progress within 6 to 12 months.
Okay, excellent. Thank you. And we have time for one more question here before we leave. This last question asks, can you elaborate on the key differences between clinical trials and routine practice when it comes to MRD assessment?
There shouldn't be. I think that, again, providing that there is progressive access to the new therapies in the routine setting, there will be more and more patients achieving complete remission, and therefore there should be more and more interest in performing MRD assessment. And one critical aspect that I would like to highlight is that we should not judge on the utility of a biomarker, be it cytogenetics, MRD, beta-2M, albumin, whatever, based on guidance for treatment decisions. Biomarkers are not performed only for treatment decisions. They are also performed for patient prognostication and for having greater knowledge about tumor biology. Therefore, I think that MRD should be performed in routine practice as well as in clinical trials, ideally using the same methodology that is commonly used in clinical trials, which is that endorsed by the International Myeloma Working Group.
Okay. Thank you for your time today and your important research. We would also like to thank LabRoots and our sponsor, BD Biosciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Thank you. Bye-bye.